Good morning. Good morning. All right, that's better. So Dr. Lynch and Dr. Payne and I would like to go through, a really broad overview of Friedreich's ataxia to start off the day, and right at the beginning also tie that to the direction for therapeutic intervention. And the way that we're going to do that is I'm going to start by talking about the genetics, and then Dr. Payne's going to talk about the cellular pathways and about the protein frataxin. And then Dr. Lynch is going to specifically tie all of that together with the treatment pipeline that many of you have probably seen, and what therapies are under development and how they tie back to the cellular pathways and the genetics. So I think we all know some of the basics, but just to start off at the beginning, Friedreich's ataxia is a genetic inherited condition, and that was known long before the gene was identified. Actually, when Dr. Friedreich first described the condition, he had already suspected that it was inherited based on family history information. Friedreich's ataxia is considered a rare disease. It affects about 1 in 50,000 people, and about 1 in 100 are carriers, and I'll explain what that means. If 2 people are carriers, that's the only way somebody inherits Friedreich's ataxia. So 2 people are carriers, then their offspring have a 1 in 4 risk. So the gene for Friedreich's ataxia is on chromosome number 9. And there are hundreds of genes in that little area where that yellow arrow is, but in that little yellow arrow area is where the gene is. And the gene was identified back in 1996. And it was identified by several different groups doing family studies that had actually linked to this area of the chromosome, and then they kind of narrowed in on the gene. So the first thing I want to make sure everybody knows is that we all have the gene for Friedreich's ataxia. Everyone has the FA gene. What matters is it coded correctly? So this is just a little schematic of the gene. And our genes are divided up into regions called exons and introns. And the exons are the coding regions of the gene, and introns are areas that contain DNA, but actually as the gene gets read or transcribed, those introns get cut out in order to make the protein. Is this a pointer? Okay. So when the Friedreich's ataxia gene gets read or transcribed, all these little introns get cut out, and you get a piece of mRNA, which then gets translated into the protein. So we all have the Friedreich's ataxia gene, like I said, and what matters is the DNA throughout the gene coded correctly. In the intron number 1, there are what's called triplet repeats, and this is where DNA sequence repeats itself over and over again, and this is fairly common, not just in the FA gene, but in other parts of our genome. GAA is what a triplet repeat means, and there are other type of genetic repeats that also exist throughout our genome. But what was identified in Friedreich's ataxia is that the number of GAA repeats was abnormally long. So most people have about 3 to 30 GAA repeats sitting in this spot. And what they found in people with Friedreich's ataxia is that they could have anywhere from 100 to several thousand of these GAA repeats. So most people with FA, both copies of their FRDA gene, and I forgot to mention this, but we not only all have the FRDA gene, but we have 2 copies, one that we inherit from mom and the other that we inherit from dad. And most people, 95% of people with FA, have too many GAA repeats sitting in this part of the gene, and usually anywhere from 66 to 1,700 is what's been reported. The other 5% of people with FA, their genetic mutations are slightly different. So on one gene, they have expanded GAA repeats, but elsewhere in the gene, in these exons, they have what are called point mutations, where a single piece of DNA might be miscoded. And these are just some common point mutations that several people have been reported with. So pretty early on, after the gene was identified, what was noticed was that the longer the triplet repeat, the earlier the age of onset somebody had a diagnosis. So here in this graph along the bottom is the length of the triplet repeat, and it's actually the smaller of the two. Remember, you have two genes, so it's the smaller of the two triplet repeats helps predict the age of onset. We all know that Friedreich's ataxia is slightly different in everybody. There's some of that variability is going to be based on the length of the triplet repeat, but we can't explain all of the variability based on the triplet repeat. Sometimes people with point mutations also have other features more specifically. For example, the optic atrophy is more common in people with point mutations. Some different features and neurological symptoms can be seen as well. Many of you have actually participated in this study. Anybody who's done cheek swabs and blood samples over the past few years has contributed to this, and I wanted to make sure to share it with you. But what we've been doing is we've been measuring frataxin. So over here is the amount of frataxin, and this is the length of triplet repeat, again, down here. And what you see is that, in general, the longer your triplet repeat, the less frataxin protein you produce. And this is another way to look at it a little bit easier, but we've been collecting frataxin on everybody, not just people with FA, but also on general people from the population. And you can see that what we call controls have, let's say, nearly normal or 100% frataxin. People who are carriers make about 50% frataxin, and individuals with FA make anywhere from a few percent of frataxin up to about 20%. The good news in this is that carriers don't have symptoms of FA. And so as we think about therapies, we don't need to get the frataxin all the way up to here. We only need to get the frataxin, hopefully, up to here. And that's incredibly important. The other thing that's incredibly important is that some frataxin is getting made. So why is some frataxin getting made but not enough? So if we go back to the gene, what happens is as the gene gets read, I mentioned earlier these introns get cut out, and the gene gets read and you get the protein frataxin at the end. Well, when these triplet repeats are sitting here, the transcription doesn't happen as it should. It's not as efficient. It's blocked, and we don't get as much frataxin. So you can sort of think of the gene as being turned off or being silenced. So you might ask, well, if the mutation's sitting in this intron, you've told me that that's a non-coding part of the gene. If the mutation's in an intron, why does it cause so many problems? It's in a part of the gene that doesn't code for the protein. In order to explain that, I want to back up a minute and first remind you that our chromosomes, that chromosome 9 that I showed you before, has thousands and thousands of genes in it. And what happens is our DNA has to get condensed. It needs to get tightly packaged. And the way that the DNA gets packaged is it gets wrapped around these proteins called histones. and that's what forms these nucleosomes. And it's this whole packaging process to condense all the genetic material. And this condensed structure is called chromatin, and it makes up your chromosome. Well, there has to be a way for this to get turned on and off, like for the chromatin to get opened up so that the gene can get read, and when the gene doesn't need to be read, it needs to be packaged back up again. Oh. So Dr. Wells initially described that what happens when there's an expanded repeat here is that you develop this sticky structure. He called it sticky DNA. But basically, the gene wants to stay all folded up and condensed, okay? It doesn't like to open, and that's part of that silencing process. So the DNA likes to stay in that super coiled, condensed state. So in order to get the DNA to open naturally, even without the triplet repeat, there are proteins and signals that happen to turn genes on and off. And one particular type of protein and signaling method that we've heard a lot about, and we're going to continue to hear a lot about, are histone acetylases and histone deacetylases. So histone acetylases help signal opening of this chromatin. And histone acetylases, we can just call them HATs. So HATs help open the DNA and allow the gene to be on. Histone deacetylases lead to the packaging signal and turn the gene off. So HDAC inhibitors are inhibitors of the signal to package the DNA. And by introducing one of these histone deacetylase inhibitors, you're encouraging the gene to be open and on more than it otherwise would. So in the 15 years now since we identified the gene, we're now starting to get to the point where we not only understand the genetic underpinnings, but we're understanding them enough to be able to go in and devise specific therapies to target this to target Friedreich's from a genetics perspective. Histone deacetylases are one way to do that. There will be others, and these are called epigenetic modifications, and epigenetic modifications are going to be especially helpful in Friedreich's because that mutation is in the intron. We don't have to actually fix the coding part of the gene, and that's incredibly encouraging. If we had to fix the coding part of the gene, that would introduce additional challenges. So there will be other epigenetic approaches that come along that try and help keep the gene open and get more transcription. At this point, what I'd like to do is hand it over to Dr. Payne so that he can kind of pick up from the genetics and then lead you into the protein and the cellular mechanisms.